right, so today we're going to be talking about exome sequencing. I have nothing to disclose for this talk. And the reason I want to talk about exome sequencing is really there's sort of two announcements. Obviously, as many of you know, the departments of laboratory medicine and pathology have now merged into a single department. And with that, there's been some changes in the genetics laboratory. So historically, there's been two different genetic laboratories, genetics and solid tumor laboratory based in laboratory medicine, and the Northwest Clinical Genomics Laboratory based in pathology. And now with our merged departments, these labs have now merged into one. So, I've been here about 6 months, and it's been an exciting experience to see really the strengths of these two laboratories come together. And the other announcement is that we've been working closely over the last few months on a partnership with Seattle Children's Hospital, really in an effort to tailor our exome sequencing service to the needs of Seattle Children's and provide the best possible exome sequencing to the patients. So as of last week, patients at Seattle Children's who are getting exome sequencing are now going to be sequenced here. And this is really a great partnership between these two institutions that will be quite helpful for both. All right, before I talk about exomes, probably good to revisit gene panels. So gene panels have been around far longer than exome sequencing. The typical scenario will be a clinical team will evaluate a patient. In this example, the patient has a broad neck, short stature, pulmonary valvular stenosis, intellectual disability. And so in this case, the team may be suspicious of a disorder called Noonan syndrome and want to order a Noonan syndrome gene panel to evaluate if this is in fact what's going on in this patient. And of course, those of us in the clinical laboratory know that a lot goes on behind the scenes when we're developing tests. So for example, if our group was developing this Noonan syndrome gene panel, we would do a lot of research. We'd find what are the genes associated to Noonan syndrome. So in this case, there's about 9 genes. We might also consider potential differential diagnoses of Noonan syndrome. So Noonan syndrome, the genes involved in Noonan syndrome are part of the RAS-MAP kinase pathway, and there are other disorders that have a lot of phenotypic overlap with Noonan syndrome. So it makes a lot of sense to include these genes on our panel as covering part of the differential. And while developing this test, we go through a process of optimizing, testing its performance, seeing are there any critical regions important for diagnosing patients that are maybe not performing so well on our test, and maybe we need to sort of tweak our test a little bit to make sure we really have the sort of highest diagnostic yield we can possibly obtain through sort of the technologies that we have available to us. And then, of course, through the process of offering this test, we're going to generate a lot of expertise, both clinically and genetically, simply by seeing patients and seeing genetic variants and getting a lot of familiarity with these genes. So, that's really what's going on behind the scenes when these gene panels get ordered. But it's not always as straightforward. So here's an example. Clinical team evaluates a patient who has intellectual disability, developmental delay, autism spectrum disorder, and has dysmorphic features. So this could represent a really wide range of potential diagnoses. So which gene panel should this team order? Where did they start? Maybe they have some idea, but it's a little bit of a guess at this point. And so this isn't really representing a distinct recognizable genetic condition. Another thing that could happen is if we revisit the panel patient that we just covered, what if that Noonan syndrome panel test came back and it was negative or uninformative? So no disease-causing variants that were identified. What's the next step there? What should they be ordering? And is a gene panel really the next best option? So we'll come back to these. But first I want to just give a quick refresher on the human genome. So our human genome is quite big, about 3 billion base pairs, and about 20,000 or so protein coding genes, and this is what we call the exome. So of course, when we're talking about exome sequencing, that's really what we're focusing on, are these protein coding genes. And our exome spans about 180,000 exons, and even though this is quite a lot, collectively this is really a small fraction of our genome overall, so it's only about 1.5% of the genome. But of course, our proteins are extremely important to the function of all of us. They have many, many roles: structural, regulatory, signaling. There's just really a huge list of kind of functions of proteins in our bodies. And if this isn't fascinating enough, of course, we know that the genome itself is also quite complex, and even more complex than shown here. But most genes do exist with multiple exons. and through the process of alternative splicing, we're able to generate multiple different protein isoforms from a single gene. So really a huge amount of information packed within our genome. For Mendelian disorders, which are disorders, really here I'm talking about disorders in the germline, so have the potential to be inherited in the next generation. There's been over 8,000 described. Individually, many of these disorders are extremely rare, but collectively they're quite common, can affect millions of individuals and families per year. And I looked a couple days ago in Exome, which is a database of kind of cataloging known human gene disease associations. And currently there's about 6,700 disorders where we actually know the molecular basis of the condition. And about 4,000, a little over 4,000 genes that are associated to one or more of these conditions. So that means that about 2/3 of the genes that we know are associated with Mendelian disorder are only associated with really one sort of Mendelian disease phenotype. But about 1/3 of them are actually associated with multiple potential Mendelian disorders. And so here on the right is just an example of the LMNA gene. You can see that it has multiple different potential disease associations. Some of these are dominant, meaning that you have 2 chromosomes from each of your parents. So if only one chromosome is affected, that's what we call a dominant disorder when that's sufficient to cause disease. So something like dilated cardiomyopathy, progeria. There are other conditions that are recessive in this gene where you need actually both copies from both your parents to be impacted in order to cause disease. So a particular type of Charcot-Marie Tooth disease. And then you can see there's other disorders like Emery-Dreifuss muscular dystrophy type 2 here, which follows both dominant or recessive inheritance. And likely a biased number, but it's thought that about 85% of disease-causing variants occur in our exome. And I say that's biased because, to be honest, it's just a lot easier to interpret variants in these protein coding regions. If they impact protein, I can at least make some assumptions about the potential function of that. Does it cause loss of function? Is it a missense change in an important domain? It's a lot harder to interpret variants outside of the exome or outside of these protein coding regions. We simply just need a lot more information to understand those. So majority of variants are occurring in the exome, but again, I do expect this to change over time. So if we go back to the 2 scenarios I gave before, hopefully it's a little bit more clear now that exome sequencing might be a great test for these 2 scenarios. So in one where it's not necessarily a recognizable specific genetic condition, looking at the entire exome would be a great way to potentially find a diagnosis in this patient. Or once you've exhausted sort of more traditional approaches, at this point we can look much more broadly and hopefully find some kind of answer. So of course I'm a laboratory director, so when I think about testing, I'm often thinking about how we build tests and really tailoring tests towards the needs of the patients and the patient population that we serve. And exome sequencing, really there's a lot to factor in here. So the patient population we might test can include any age. I've tested and reviewed data from patients that were 1-year-old babies in the NICU to adults who were grandparents. Our test has to be designed to fit any kind of family configuration. So in exome sequencing, it's because this test is so broad, it can be quite helpful to include other family members in the test. And these can increase the diagnostic yield. So for example, a trio analysis where we test a child and both of their parents, we can actually interpret or gain some idea of the significance of variants, whether or not they were inherited and how they were inherited from their parents. And then any number of other family members. So, our test now has to fit from what could be just in one individual, a proband-only analysis, up to any number of additional family members. Our test has to cover disorders that really fit within any mode of inheritance. We might see disorders that are dominant. We might see disorders that are recessive or X-linked. Some of these variants will occur de novo, others will be inherited. We have to be able to cover all of these scenarios because all of them at some point will occur. We need to be able to pick up basically any phenotype. Patients who come through for exome sequencing can have any number of the body systems affected. Sometimes and quite often there's multiple systems affected. And again, we're trying to really identify any kind of Mendelian disease that can be picked up by our test. Our test has to be designed to pick up any number of range of variant types, at least those that, again, are within the limitations of the short-read next-generation sequencing. So this includes single nucleotide variants, small indels. These variants could occur in coding regions. They might also occur in non-coding. Of course, we call exome sequencing targeting the protein coding regions, but in reality, we do sequence into the introns and may target other non-coding areas that are known to be clinically relevant. So we do pick up non-coding variants in this test as well. And then it's a little bit more challenging in exome, but I think we're moving in the direction that we can call single exon losses and gains through exome sequencing as well. And finally, I don't have time to talk about it today, but I just want to touch on it that there are kind of additional flavors of exome sequencing. One of them is prenatal exome sequencing. As you can imagine, there's certainly some additional considerations in this type of scenario, as well as critical care exome sequencing. So you may have sort of heard of the news or sort of other places of sort of rapid testing performed in critically ill patients in the NICU or PICU or even in adults. But again, these have additional considerations, so for the purpose of this talk, I'm not going to cover these. All right, so when we do exome sequencing, what do we find? The answer is find a lot. Each of us carries about 25 to 30,000 protein coding variants. So that's a lot to go through. And this graph here is just showing for various superpopulations, really going through the number and breakdown of the variant types. So you can see here different populations, and then on the left for each column is the number of heterozygous changes, and on the right is the number of homozygous for the variant allele positions. So if we scan across here, we have a lot of variation. We have a lot of missense variants. That's not too surprising. We can use tools like SIFT or polyphen to find, to give some kind of prediction whether or not these variants are predicted to be damaging. And as you can see, we still have a lot of those. Even if we look at variants that are generally predicted to be quite deleterious, things like frame shift or nonsense variants, as you see, we have a lot of those too. So, how do we do this? This really is, this test might identify up to 90,000 variants. And what we're ultimately going to report out is one or maybe two variants that we think are causing the patient's or contributing to the patient's features. So this really is the needle in a haystack scenario. My goal is to find of 90,000 variants, that one or two, maybe three, that are going to ultimately get reported and hopefully help the care of this patient. So I wanted to walk through that step a little bit because, this really is a complicated process that I think it's worth having some understanding of. So one of the first things that we do when we're analyzing exomes is take those 90,000 or so variants we've identified and bring it down to something that's manageable. So, this 90,000 includes our 25 to 30,000 protein coding variants, a lot of intronic variants. We also have a lot of false positive calls in there. Of course, we want to optimize our ability to detect variants, so that does mean we do have to deal with false positives in our data set. So how we get through this is to give some kind of prioritization. So, pulling out variants that have some kind of characteristic that might make them a little bit more likely to be informative for the purpose of our test. So things we might filter on or prioritize based on is rarity. If most of these diseases, as I said, are really quite rare. So, a disorder that affects 1 in a million, if I see a variant in that gene at 10% of the population, it's simply too common. It just doesn't fit with what's known for this disease. So those are things that I really, if I'm trying to sort of use my resources wisely, is not really a good place to put my time. So we'll look at allele frequencies, and there's a lot of databases like Nomad out there that have very large data sets of people and populations to use for this purpose. Another thing is, has the variant been seen associated with disease before? So there's a lot of disease databases we can take advantage of, like ClinVar or the Human Gene Mutation Database. We can look at things like what's the effect of the variant if it's protein coding. Does it cause a nonsense variant? Does it impact splicing? Is there anything else about it that, like its prediction of deleteriousness, that I can use to my advantage to help pick out the ones that seem more interesting to review further? I'll look at zygosity. So if I have a rare homozygous variant, that gets my attention. If it's a male and it's a hemizygous variant, that gets my attention. We look at inheritance when I have that available. Obviously de novo variants are quite rare, so we look quite closely at all of those. But is there a compound heterozygous variant? So something that's been inherited, two different variants inherited from each parent in the same gene. This is a gene that the variant's been identified and even associated to disease. If I have limited time and resources, probably less useful for me to spend time on genes that are not known to cause disease than those that are. I'll look at the patient's features. So there's a lot of really interesting databases out there. One of them is called the Human Phenotype Ontology, which I've shown here, that tries to link genes with specific features rather than a necessarily overall disorder. So you can use that to your advantage to help prioritize variants you want to look at. And then finally, quality. Obviously, if a really low quality variant's been seen before, those are ones I want to look at. But again, probably most of these are going to turn out to be false positives. So that's sort of another feature I might look at to help make these decisions. And to try to standardize this process, we sort of put all these elements together into some kind of workflow or a flow chart here to again help bring the number of variants we review down to something that's actually manageable. I'd love to look at 90,000 variants, but the turnaround time for this test would be quite too long if that was the case. Okay, so we've brought our variant list down to something we're going to review. So now the process is to really go one by one and assess them, each of them individually. And there's a lot of things I'm thinking about when I look at a variant. One of them seems pretty obvious. What's the phenotypic fit? So I've identified a variant, it's in a gene associated to disease. How closely does that disease overlap with the patient's features? Sometimes you have a really good fit, sometimes there's no overlap at all. Sometimes the overlap may or may not be a specific overlap. So for example, something like short stature, if that's the only shared characteristic between my patient and a given disorder, that's not as informative as something like anophthalmia, where you actually have no eyes. That's a very specific feature. So regardless of whether or not my patient had every feature of that disorder, the specificity of that phenotype would certainly get my attention. And then of course I do always factor in age. So, patients, as I said, I've tested babies before and they may not have developed or reached an age in which they would develop some certain kind of characteristic features of disease. So I always sort of keep that in the back of my mind. Another thing I look at is the inheritance. So, we find these variants when we have inheritance available to us. It can be very helpful. So, does the story fit is kind of what I think of when I come across this. So if it's an autosomal dominant disorder, did we identify a de novo change? If it's recessive, do I have two alleles in the same gene? If it's X-linked recessive, did I identify it in a male? Those are much more, the stories sort of fitting together. I sort of have everything I need to explain this result. Versus when, it's an autosomal dominant disorder, but it's inherited from a healthy parent, or it's recessive and I only have one allele, or an X-linked recessive disorder in a female. The final thing that I tend to put some emphasis on is variant pathogenicity. So, regardless of whether it's coming from my patient or elsewhere in the literature, is there evidence that this variant is disease-causing or not? So pathogenic versus benign. And the truth is, obviously, it's pretty easy when we sort of land all on one side or the other. So, variants that are really good fit, de novo, been seen before, the pathogenic, those are really easy to pick out and report. And similarly, if it doesn't overlap at all, it's too common, those are things that are quite easy to rule out. But it's not that simple. And I do factor all three of these together when I'm trying to decide whether variant is sort of relevant for reporting. So for example, I might have something that's not a very good fit, but if it's de novo and it's pathogenic, I'm going to look really closely to make sure that I evaluate that phenotype really closely and don't necessarily rule it out simply because it's not the best fit sort of at first glance. So let's actually give this a try and sort of see what it feels like to be in my shoes analyzing these variants. So let's say we have a situation where we have identified two variants in a patient in the same gene. So this is great. It's an autosomal recessive phenotype. We've got our two variants, so our expected inheritance, the story is sort of fitting there. And let's say one of these is a loss of function variant, the other is a rare missense variant. So the loss of function variant, those aren't necessarily always pathogenic, but in this case, we will say it is. So we're kind of in the middle of the road. One of these variants seems quite suspicious, and the other one we're just not sure about. But then we look at the phenotypic overlap, and it's not that great. Maybe just a couple features, relatively nonspecific overlap with the disease we've identified that's associated with this gene. Do we report it or not? I don't know. That's a little bit of a judgment call at this point. What about another scenario? Here where we have, maybe it's a dominant disorder. We've identified a VUS, it's a rare missense variant. It's inherited though. So, inherited from an unaffected parent. So, that sort of makes it a little bit less suspicious. But the gene is associated with disease, which really does have some overlap with our patient. Not really a home run, but certainly some overlap. What should we do in that scenario? So these are really the challenges that we face every time we go through exomes. We run into these things that, I can't rule out. There's issues of phenotypic expansion. So kind of historically, we've known and characterized disorders based on their phenotype alone and not necessarily genetic information. So phenotypically, it's easier to put sort of patients at the severe end of the spectrum to recognize them as the same or having the same condition. But as we've done more genetic testing, we've sort of seen this, the phenotype broaden. And so that's something I always have to keep in the back of my mind. Similarly, there's reduced penetrance of some disorders. So in this case here, where it's inherited from an unaffected parent, doesn't necessarily mean I can conclusively rule it out, but certainly does give me pause. And there's really not a good answer here. It does end up being a case-by-case basis. And we do have guidelines out there to help standardize this within the laboratory. But at the end of the day, it always comes down to the sort of the situation at hand and evaluating this at a case-by-case basis. The other part of this is then trying to decide, in these sort of situations, what do we report? In a gene panel setting, it's a much more targeted analysis. You're looking at far fewer genes. All of your genes you're looking at should, at least in theory, be somewhat related to the patient's indication for testing. So maybe, the point where you start reporting is the point of where you can't really rule things out. These things, rare missense variants, for example, are a great example of things that we see. They could be involved, they could be just sort of benign bystanders hanging out in your genome. We have a lot of rare variations, so just because you have one doesn't necessarily mean it's disease-causing. The problem is this doesn't scale when we start talking about exome sequencing, because we just have there's just a lot out there, there's a lot of variation. So for me, there's a lot I see I can't rule out. So I sort of shift that line of where I start thinking about reporting into the domain of things that, not necessarily that I can't rule out, but things that I actually have some suspicion of. And that's the point where we will consider reporting. So reportability does vary by labs, but this is sort of the approach that I've been taking just to, we don't want to report back, here's 50 variants we couldn't rule out. Obviously, I don't think any of our ordering providers would be able to sort of get through that in any kind of useful fashion. So something else we've discovered as we've done exome sequencing is that some patients don't just have a single genetic disorder, but might have two or more. So this was a study from Ambry where they looked at about 7,500 patients that had undergone clinical exome sequencing in their laboratory, and they found that 2% of their patients had two or more genetic Mendelian disorders. And this has been pretty well described. And so this is kind of another example of the patient population that is really well suited for exome sequencing. Of course, when these patients are seen clinically, they're not recognized as having a single genetic disorder because, in fact, they don't. They have two or three. And these have blended together to sort of create a unique phenotype in that patient. So these are sorts of things that we can pick up on exome sequencing that would be quite difficult to deal with otherwise. Another situation that we encounter in the world of exome sequencing is kind of secondary or incidental findings. So these are medically actionable findings unrelated to the reason of testing. So this isn't necessarily unique to genetics. Certainly our colleagues in radiology encounter this. Say you have a patient who had a shoulder injury, they go have an x-ray and a tumor is identified. That will be followed up on. So a similar scenario, but in our world it is a little bit different. So in our case, our patient that we looked at earlier with intellectual disability, developmental delay, autism, and dysmorphic features, has exome sequencing performed, trio exome sequencing, and then we incidentally encounter a disease-causing variant in the BRCA1 gene associated with hereditary breast and ovarian cancer. So what do we do at this point? It's obviously going to be very useful for the family to have this information. But again, the point of this test was to identify this patient here, patient 10 with the arrow pointing here. So, to identify the cause of their features, this is an unexpected result. You can appreciate that we included the family members. This was trio exome sequencing. So not only are we getting the result here on our patient, we're also getting the result on the patient's parents who are part of the test. So that information would be returned back to them. Again, a very unexpected result for them. And then you can see, we all have, there's families at play that we also have to consider. So these individuals weren't necessarily part of the test, but some degree of cascade testing may be pursued in order to determine who else in the family may have inherited the same variant. So these situations are pretty difficult in the lab to figure out, when have we found something that's sort of meeting this threshold of medical actionability to report back. But thankfully, we've gotten some guidance from the American College of Medical Genetics on defining disorders that should be resulted back. In this process, the patients do get a say in this. So the consent process for Exome is whether or not they want to receive these types of results. So they do have the option to decline or opt in. And I call these secondary findings rather than incidental findings because we will go through and actually search these genes. This is now part of the test. So it's not just that we incidentally run into them. We actually will look quite closely and see if there's anything potentially disease-causing within these genes and report them back if they're seen. And the majority of these disorders are the cancer susceptibility genes. There's several sort of cardiac disorders like arrhythmia or cardiomyopathy. These types of disorders can predispose you to sudden death, so it's obviously quite useful to have this information and be able to manage the condition sort of prior to what can be the presenting feature of sudden death. And there's several other disorders here, which I encourage you to look at if you're interested. The problem is that's not the only thing we see that's a bit unexpected in the exome. We're looking at everything, so we find everything. So there's a few things we have to be prepared and know how we're going to deal with these situations. One of them is non-paternity. We do a lot of trio testing. Again, this does increase diagnostic yield, so it is encouraged. Sometimes we identify that family relationships aren't at what are expected. So we have to have policies in place to deal with this. This is reported back since obviously it does impact our ability to perform a trio analysis. But as you can imagine, it is quite a challenging scenario to deal with. Other things we might see, we might find that the patient's a carrier of a disease that's really not related to what we're looking for. Maybe our patient from before was found to be a carrier of cystic fibrosis. Maybe they have beta thalassemia trait or any other number of rare autosomal recessive Mendelian disorders. So these are things we see. Other things we might run into are adult onset, kind of quote unquote non-actionable findings. Amyloid precursor protein causing early onset Alzheimer's. What if we find a disease-causing variant there? Or what about the ApoE gene and the risk allele for late onset Alzheimer's? What should we do when we come across these? We find pharmacogenetic results. I've found a patient where there was dihydropyrimidine dehydrogenase deficiency. So when you have this condition, it's associated with a life-threatening toxicity when exposed to 5-fluorouracil chemotherapy. So it's a very commonly used chemotherapeutic. And it's not necessarily always standard of care to test for this prior to receiving this chemotherapeutic. So what should I do ethically when I run into this sort of situation, exome testing, particularly in a child? Other examples are things of sort of uncertain or unclear actionability. So these are all examples I've run into in the past. So we see low penetrance cancer genes. What should be done then? Or if there's a cancer susceptibility gene, but there's no guidelines for changes in management once that's identified, how should we handle those? So an example I've come across is a variant in MID-F, which increases your risk of melanoma. It's a relatively modest increase, but it does seem quite believable. Another example is there's phenotypes that have really variable expressivity. So I've encountered a disease-causing variant in variegate porphyria. So this features can range from, a lot of people are completely asymptomatic to cutaneous manifestations and life-threatening acute neurovascular attacks. So what should I do in that scenario? Most patients are asymptomatic, but it does have a potentially life-threatening outcome, and obviously is quite difficult to diagnose, so it could be quite useful to know. Another example I've encountered in the past is, so we had a patient who was a child, female child. She had X-linked glucose-6-phosphate dehydrogenase deficiency. So again, this is X-linked, but since we'd done trio testing, we actually knew it was her dad. It was who she inherited it from. So again, many of these patients are largely asymptomatic, but certain exposures can trigger this life-threatening episodes of acute hemolytic anemia. So what should we do in those scenarios? And now it's not even really our patient we're concerned about, it's actually our patient's father. So I certainly don't claim to be an expert on sort of dealing with these sort of scenarios, but I just wanted to highlight just how diverse the results you might come across are, and kind of the difficulty in trying to figure out what's best to do. So in many of these scenarios, probably spend a whole lecture sort of discussing each of these, and certainly for the adult onset disorders, many of these patients are children, so there's certainly something to be said about that child's autonomy and given the chance to grow up and decide whether or not they themselves want to receive this kind of information. But certainly those sort of pharmacogenomic or unclear actionability results are ones that I've found particularly challenging to deal with. And for those reasons, as I said, it does vary by lab, but we tend to rely more on the guidelines from communities like ACMG to sort of decide what the best practice should be when we run into these situations in an exome setting. Okay, so now that you have a taste for just how complicated this process is, we've had our test ordered, our lab has gone through and sequenced and bioinformatically processed, which I didn't even really go through for the purpose of this talk. We identify a lot of variants. We have to go through and decide what we're even going to look at because there's too many to really look at. And then comes the process of review. And hopefully now, as I said, you see how complicated this is, both in terms of deciding whether or not variants should be reported, whether they're related to the indication for testing or some other sort of situation. So here at UW, we use an integrated clinical reporting model where we have experts from various disciplines in our group. But for exome testing, it includes it includes individuals from both the University of Washington as well as Seattle Children's Hospital to really guide and take advantage of the expertise of all of us collectively to make the best decisions we can for what's best to report and return back to the clinician. So this is something that a lot of other labs don't actually do, and I've been very pleased to be part of this process here at UW because I do think it really does provide the best opportunity to give the best care to patients. Okay, so something else that I think is always important for any kind of test is to know and understand its limitations. So I wanted to spend a little bit of time going through that. Exome sequencing is done by short-read sequencing, and there are some limitations of short-read sequencing that really apply across the board. So some variants we just can't detect by short-read sequencing. So for example, the triplet repeat expansion disorder. So we're not going to pick up something like Fragile X syndrome or Huntington's disease. This is really just outside of the scope and ability of our test to identify. Similarly, there's going to be other regions we just don't get good data for. Maybe there's low coverage, or maybe there's issues with mapping, and we can't uniquely sequence reads from those regions. And this can be important to think about. So here's an example. So this is the gene stereocillin. So stereocillin is one of the most common causes of mild to moderate hearing loss. And stereocillin has this very challenging, highly homologous pseudogene. So this is a copy of the gene that has sort of become non-functional elsewhere in the genome. So if you look at stereocillin and you look at its pseudogene, about 98.9% of it, including the introns, is identical. And in the first half of the gene, it's actually 100% identical. So if I was testing a patient with hearing loss with exome sequencing, how would this area perform? The answer is not very well. So we can see here, this is just showing mapping quality on the bottom of the various regions of stereocillin. And so as you can see, the first half of the gene is really just, that identity problem really prevents us from being able to look at this. And stereocillin is just one example. There are numerous examples of problematic genes. But again, this is such a broad test that I can't go back and fill all of these in. So, if I have a patient with hearing loss, I have to consider that I'm likely going to miss this and need to keep that in mind. The case of gene panels, I think, if we're designing a gene panel for hearing loss, we're gonna know about this, and we're likely gonna probably do something to make sure that we overcome the ability or the limitation of sequencing in this region. So kind of one of the differences between exomes and panels. Another thing to think about is that exomes, we don't necessarily get the highest coverage in exome, and I'll tell you why in a moment, but that means that we have to think about what kind of variant type we're expecting. So exomes may not be the best for conditions that have low-level mosaicism or somatic variants, and that really comes down to cost. It's not that it can't be done, it's just that it costs too much. So we've seen that the cost of sequencing has dropped pretty dramatically over time. So this is a graph. So you can see on the x-axis is over years, and on the y-axis is the cost per raw megabase of DNA sequence. In 2007, we saw kind of a pretty drastic drop, but over the last couple of years, that's really sort of stabilized. And the cost really comes down to sort of, there's two elements at play here. One is the size of the region you're evaluating. So obviously we're going to require more data to sequence 100 genes than we are just a single gene. But the other element of this is how deeply we sequence each of those genes. So, and by coverage, how many times have we sequence the same base, which you would need to do for example, in a mosaic or somatic condition where maybe only 1 to 2% of cells might have a particular variant while you're testing. So what happens is that for panels or sort of smaller targeted regions, you're starting with a lot less, so you can sequence that smaller amount at much higher coverage with a sort of more minimal increase in costs. So you can see here the slope of that blue line is a lot less than when we start looking at much bigger tests like exomes or genomes, where in order to reach those really high coverage, because we're just sequencing so much to begin with, it's a much, the growth and cost of it increases much more dramatically. So, our exome test, we get about 100x coverage. This is more than sufficient for the, most of the patients which are going to be Mendelian disorders, germline disorders. But that does mean that for conditions like somatic or mosaic changes, we do have more limited ability to detect those. All right. So as I said, exome sequencing, we're targeting our 20,000 genes. This has a lot of advantages. It's disease agnostic. We can really accept a wide range of patients and try to get them an answer. And for many of these patients, there's really no other option. So this is a great test to have. So even though it is incomplete, it's very broad. And I want to just contrast this again with our panels. Here we're targeting a much more limited, focused number of genes. Because this is so focused, these panels tend to be designed more towards high performance of specific genes. So that example of stereosilin that I gave, which didn't perform well by exome, certainly we would go and do maybe supplemental assays or tweak our capture to make sure that we covered that gene and sequenced it thoroughly. You develop content knowledge in gene panels, which is just really not feasible by exome because, of course, we're really sampling all Mendelian disorders. So even though the analysis of a panel is much more limited, it is very focused. And so really that's kind of in my mind how I think of exomes and panels, of exomes being much more comprehensive, but panels being a bit more complete. So I can remember back to my training, hearing someone ask, why would we even use panels? Why don't we just order exomes on everyone? And hopefully it's clear now that I do still think panels do have their place and their use, even though exomes are also often quite useful as well. Okay, so how does exome actually perform? What's the utility of it? So this is just an example I'm going to go through because I think it kind of illustrates what we see out there. So this was a study looking at a cohort of intellectual disability. They identified a disease-causing variant in about 30% of the patients, and this is pretty normal or kind of a typical range we'd expect to see in exome sequencing. It varies by what kind of patients you're testing, and whether they've had previous genetic testing or not, and whether it's a trio analysis or proband only. But at the end of the day, it usually is around 30% is our diagnostic yield. However, these patients that receive exome sequencing often are very medically complex. The average diagnostic odyssey in this group, so the time from when they started pursuing a diagnosis until they actually received one, was on average 6.6 years. You can see in this graph showing the patients there just the different types of visits they had or different procedures, and the cost of their healthcare, which really spanned many years and tends to be really quite expensive. And in this study, they found that the traditional diagnostic trajectory averaged about $16,000. But when they used exome sequencing, that saved an average of about $3,500 per patient for those that were receiving a diagnosis. But patients who didn't receive a diagnosis also had a cost savings. At this point, we can eliminate a lot of the disorder on the differential with exome sequencing and kind of direct treatment that way. And this is true. You can look in a lot of different disease data sets or cohorts or disease domains, adults and children. This is really a kind of a growing body of evidence that exome sequencing can shorten time to diagnosis as well as be cost savings. Again, oftentimes, currently at least, exome is often performed after a lot of other genetic testing might have already been pursued when available. So there's been a lot of studies arguing that earlier implementation of exome sequencing can save even more money. All right, another thing I wanted to touch on about exome sequencing is the fact that we're sequencing all the genes. So that's really useful because our knowledge is constantly changing. We find new gene disease associations all the time. So this was a study that was performing exome sequencing on a research basis, and they found that on average, they were identifying 263 new gene disease discoveries per year. So this was averaging about 1 out of every 28 exomes they performed. There's other instances where they were able to expand known phenotypes to kind of more broader presentations. So this is great. I can look at a patient and maybe I can't diagnose their exome, but maybe tomorrow a new gene disease association will be discovered, and I'll be able to put those pieces together. So that comes into exome reanalysis. So as I mentioned, we've sequenced all the genes. We have all that data. So just because I looked once and I couldn't find an answer, I have all the data and I can revisit it and go back and potentially find something that wasn't known before. And there have been numerous studies on exome reanalysis, but I like this one in particular. So this was a study that looked at exomes that were looked at 2 different time points. So we'll start here on the left. So at the original analysis in 2012, the diagnostic yield was about 25%. In 2017, they reanalyzed those exomes, and you can see that there's a pretty big increase in the number of patients they were able to give a diagnosis to. And they looked at sort of what led to those diagnoses. So, at lower frequency, sometimes there was a new variant type, like a copy number variant that was not previously appreciated. Sometimes family studies help resolve exomes. So maybe there was affected family members. They could test segregation of the variant in the family and help resolve its significance that way. Sometimes there might just be new information about the variant from other family members. So this would cause a variant reclassification. So maybe you have a novel variant and then we find 3 other families with the same phenotype who carry the same variant. So that's going to sort of shift our understanding of the variant itself. Other times it's actually a clinical update. So maybe there's new clinical features observed in the patients that help our understanding of the patient's condition and therefore identify disease-causing variants that way. But the most common cause of finding a new diagnosis which wasn't originally appreciated was that there was a new gene disease association. And you can see on the right there is a replication of kind of a similar design. And that was true in both data sets, that most of the time when we're able to diagnose patients we couldn't before, it's because we simply have new knowledge that we didn't have at the time of the original analysis. So this is a really exciting part of exome sequencing, that we have all this data. We do the sequencing once and can go back to it and keep trying to identify the cause of these patients' conditions. Okay, so I have a couple minutes left. So I just wanted to broaden a little bit our use of exome sequencing. Of course, next generation sequencing has been an evolving landscape. And throughout this entire talk, I've really been talking about sequencing and panels and tests and really focusing more on these as clinical tests. So this is sort of what is ordered by the provider, what we're resulting back. But in my mind, I do actually split what the clinical test is from what the technical assay in the lab is. And historically, we haven't really done that because they've sort of been one and the same. If you order a panel, your technical assay in the laboratory was capture of those genes in the panel, exome to exome, genome to genome. But as we've seen over time with reduced costs of sequencing, this is starting to broaden. So it's now feasible, and there are a number of laboratories, including ours, who use exome sequencing as our technical assay. So we perform exome sequencing in the lab. This, of course, leads to our exome test, but we can also use this as the basis of our gene panel testing. So we just bioinformatically mask the data down into the genes of interest for the panel. We're not quite there yet, but I do see on the horizons of sort of repeating this process with genome sequencing instead of exome sequencing. And so these 2 scenarios on the bottom, when you see these are commonly referred to as sort of an exome or genome backbone test. So it's really sort of now differentiating what the technical assay in the lab is from the clinical test. And there's a number of advantages to doing this. So historically, it's looked like this. We've had a number of technical assays managed in the lab. We have a capture from panel A leading to our clinical test panel A. Maybe we can get lucky, and for example, on C and D here, we've combined our captures into one, so we can report out 2 assays from this capture. But you can see with exome sequencing, really simplifies operations a lot. So the assay that we're maintaining in the lab now is just exome sequencing, and we can really have a whole range of panels or other tests from using this data. So greatly simplifying our operations. Another benefit is, I think it's kind of helpful to look at cases of genes that are really traditionally evaluated by panel sequencing and what happens when we do tests like exome sequencing or genome sequencing in them. So in this example, this was a study of 46 families with hypertrophic cardiomyopathy. They had all had prior genetic testing that was inconclusive. However, when they did genome sequencing on these same families, after receiving these inconclusive results, they were able to resolve 9 or 20% of these families. And if we look closely at the types of findings they found, 4 of these ended up being in genes where we already knew they were associated to hypertrophic cardiomyopathy. However, they were really deep intronic variants impacting splicing, but they weren't targeted as part of the test. A lot of tests do target just the protein coding region, so these were missed. Similarly, there was 5 variants that were simply just in new genes that we know are associated with HCM now. However, at the time of testing, we didn't, or they weren't part of the panel. So if we use this information, I think it really highlights the other benefit of these sort of backbone tests. Now let's think of the scenario where panel B and panel D here, a new gene is discovered associated with the disorders we're testing here. So in order to update our panel, we're actually going all the way back to the wet lab. We're redesigning our technical assay, optimizing it, validating it. It takes a lot of work, takes a lot of time, it's very hard. So we're often quite slow to go and update our panels, or can be, because of these limitations. But as you can see on the right, in the same scenario, if we had all of our testing done on an exome backbone, it's really more of a bioinformatics exercise to now unmask the data, see how it performs, and go from there and see if it's feasible to add back this gene. So that's kind of the other element of these backbone tests that can really simplify operations and really help keep our panels as up to date as possible. So for the example now, and what we do in the lab currently is using an exome backbone. You can see that, I think we're moving in the direction that genome sequencing is not too far off in the horizons. And so that example I just went through for HCM, as I mentioned, several of these patients actually carried non-coding variants. So genome sequencing would allow us not only to now add back these new genes, but even add back regions that we hadn't traditionally been able to target very well, simply because it's just difficult to interpret these. But with new information, these can now be included as part of the test. All right, so quickly on genome sequencing, since I brought it up, I think it's worth just to take a moment to cover it. So tests like gene panels and exome sequencing are all targeted tests. So we take our DNA, we're able to sort of capture or enrich our DNA in the regions of interest. Maybe it's the genes we're interested in or our whole exome, and thereby reducing our DNA content that we're going to sequence down to just that region of interest. So this is really done for cost savings. As I mentioned, our exome is about 1.5% of the genome, so we're able to sequence a lot less and much more cheaply using exome sequencing compared to genomes. However, genomes a lot easier operationally. There is no capture. We take our DNA, we shear it, we do a library preparation, and we're able to sequence it directly. And genomes do have some benefit in terms of performance. So genome sequencing, you can perform a PCR-free library preparation. And this will improve a lot of the biases or kind of gaps that are introduced from PCR during sort of standard sequencing approaches. You do need more DNA, so it's not necessarily suitable for all sample types, but when you can use it, this is sort of what we see. So here on the x-axis, you can see the GC percentage, and then the coverage on the y-axis. So you can see for both exomes or genomes that use a PCR-based library prep, the coverage really drops off as we get into these higher GC content regions, but we really don't see that when we look at genomes with a PCR-free library preparation. So it's one method for us to capture regions of the genome that just have historically been challenging to evaluate. All right, so it doesn't end here. There's been a lot of really exciting advances in the last decade or so with exome sequencing, but I do believe we're at the beginning of the road, and we have a very long road ahead. One of the things that I'm always sort of thinking ahead towards is sort of what's coming next and what might that look like. And as I mentioned, exome sequencing, our diagnostic yield is about 30%. So this doesn't sound great, but these are often patients that really, they're at the end of the road. They've gone on very long diagnostic odysseys already. So even to give those 30% an answer, to me is very exciting, but we should always be striving to do better. So it's good to think about, what do we think we're likely missing in our test? and how do we get past those and improve upon that. So certainly there's some variants that we can identify currently and we just don't know what they mean. So things like exome reanalysis are ways in which we can solve those. But there's other variant types that we're not detecting or we're not currently capturing. These could be structural variants or things that were undetected by the test. As I mentioned, one of the limitations of exomes is that we don't sequence very deeply, so we're missing some of our somatic mosaic variants. And there's sort of other complex sort of situations that can arise. And as you can see on the right, there's kind of a host of upcoming technologies that I think are going to really make a big difference in trying to unravel some of these things that we can't currently interpret very well through exome sequencing. So kind of long-read sequencing, as I mentioned, whole genome sequencing. There's lots of omics technologies out there. These are all things that I think are being looked at in the research setting, and my hope is eventually enter the clinical practice and improve what we do. Taking it even a step further or a step further back, you know, I'm relatively new in my career, so I think of exome sequencing as really sort of the stepping stone into sort of a bigger world that I imagine could certainly happen within my career, of where we might be sequencing everybody as part of routine clinical care. You know, as I mentioned, our genome has a lot of information in it. We can use it for prognosis, identifying patients with disorders before they even develop symptoms, and perhaps preventing them or managing the condition better. There's really a whole host of ways we can translate the information from our genome into sort of improved clinical care. So there's that diagram on the left that sort of goes through that, you know, risk scores and drug discovery, pharmacogenomics, all those sort of things. So there's a lot we could use, but currently it's really hard to know what's the best way to do this. So it's also very exciting that here at UW is one of the centers participating in the NIH All of Us project. So this project, they're sequencing 1 million Americans from diverse backgrounds, and one of the goals is understanding how to use our genetic information and improve healthcare for everybody. So with that, I'll thank the teams, so the Northwest Clinical Genomics Laboratory, as well as the Genetics and Solid Tumor Laboratory, and our partners at Seattle Children's, and I'm happy to take any questions.
Thank you. So we have a few questions, or at least one question so far, and I have a few. So one of the questions was, when using an exome platform, can you share how you keep some of the data that patients didn't want reported or the clinicians didn't order masked? So how do you keep that data masked?
So generally for our exome backbone panels, we don't actually even generate the data, so the data is blinded to us too. So we're only really going to be looking at things that are part of the test. So that gets masked upstream of the analysis, so it doesn't hit us and it doesn't hit the clinicians or sort of downstream to the patient. So that kind of eliminates the potential challenging scenario where you might sort of see something that's not necessarily part of the test, and sort of be stuck in that difficult situation of sort of what to do with that.
Okay, thank you. So this is a great question I also had. So Sarah asks, as she said, first she says, great comprehensive talk. So you seem to make a good case for genomes over exomes. And there's talk of a one and done genome at birth as part of a newborn screening, for example. It could be reanalyzed over life. What are your thoughts about whether this idea of one and done is realistic, or if there will always be advances in platforms, captures, analyses that require consideration of getting an updated genome rather than just analyzing previously obtained data over the course of a lifetime?
Yeah, that's a great question. To be honest, part of what I think about is cost of storing data. So I know that has come up before of, we could, let's say, fast forward to however many years from now when we're all getting our genome sequencing as, maybe at birth as part of standard of care. That's a lot of data to store. So is it better, once we're going to hit a point where the cost of sequencing might actually be cheaper than storing this data over time and constantly going back to it? So that would be one of the things I would actually factor into that is actually the cost of data storage. But, we'll see how long it takes to sort of reach that inflection point.
Another question, when ordering a panel or an exome, I guess, will you automatically be revisiting the data to reanalyze it every few years?
Yeah, so that is also a great question. To me, this is a little bit complicated. Let's go through the scenario where we do kind of more of an automated reanalysis. So let's say our policy was every 2 or 3 years we're going to look at it. So that takes a lot of resources. If I find something, now I'm going back to the clinician. Is the clinician even still at my institution, or can I track them down? So that's kind of one potential variable. The other thing is if I can track down the clinician, can the clinician then track down the patient? I definitely see value in it, but I also think it can be operationally a complicated process to go through. Our policy is, we really want it to be initiated from the ordering provider. If they've seen the patient or kind of understand and have contact with the patient, that this is something that we want to pursue. And so they're the ones initiating to us that they would like to do reanalysis at this point in time. And we have policies in place of sort of what, how much time needs to go by before we'll sort of look again. But that's sort of the two sides of that coin. I definitely see the value, but I think problems can arise. So currently we are asking that the providers contact us for that reanalysis.
Thank you. I want to add a comment from our last ground round speaker. Patrick Matthias did a cost analysis of data reanalysis and clinical decision support for genomic information, and showed that storing the data and doing clinical decision support for genomic data for pharmacogenetics, let's say, over time cost thousands of dollars per assay, and that's not budgeted in at all when we do the billing for genomic analysis. So where that overhead comes from really hasn't been defined by our society. So anyways, our last ground round speaker, Patrick Matthias, could potentially chime in on that if he were here. So one more question: what are the advantages to exome over genome sequencing? I think that's already been addressed of what are the advantages over exome or genome sequencing, but I'll just add to that. What do you think would have to change for us? So what are the next steps that we'll have to change to move from exome to genome? What are the barriers that are keeping us from doing genome right now?
I think the biggest barrier is cost. Genome costs quite a bit more. Again, our exome's 1.5% of our genome, so you can imagine sequencing that is a lot less expensive than sequencing the whole genome. So that's really the biggest barrier in my mind is the sequencing cost, and that's something I expect to change in the future. So once it does, hopefully we can make the switch. I've looked at genomes in the past, and there are quite a few advantages, but again, cost is the main challenge.
I'd like to thank Jillian for coming and speaking to us and giving a really wonderful presentation. And for those of you who are online, please sign in and come back next week for our next grand rounds.
All right, thanks everyone.